Okay, so when we left off with lecture, we were talking about one way to measure heritability, which is to examine the regression of the weight of, or rather some parental trait against the offspring trait. And we were discussing what exactly was the measure of heritability, and it is the slope of the regression. And I had written on the board as r squared, which is, John Hansen was correct, that is the coefficient of determination rather than the slope. The slope is actually expressed as beta. Some people were talking about m, which is normally the slope of a line of that equation, but for regressions, it's actually a different equation where it's actually, the m is sort of, you can think of it as substituted for the Greek symbol beta. At any rate, so beta is the heritability, slope of the line of the regression, and so I wanted to mention a couple things about heritability that is difficult for people to wrap their heads around, and even people who do this for a living. So it's important to remember that heritability is the proportion of phenotypic variance explained by genetic variance, and specifically by genetic variance. And then we can break that down into additive genetic variance versus general genetic variance. It is not the same thing as heritable. So I want to draw the distinction between heritability, which is h squared, either little h or big H squared. It doesn't not mean the same thing about as saying a trait is heritable. So that is to say, it's not the same thing as the degree to which a trait is genetically determined. So, for example, if you have a population where heritability is essentially 0, that means that all of the phenotypic variation for a certain trait is due only to environmental variation. And if you have no genetic variation in the population, let's say they're all genetically identical, they're clones, that's true of certain animals and plants, heritability of a trait in that group of individuals is going to be 0. That isn't to say that that trait is not genetically determined. It is. But the proportion of variance of the phenotype of that certain trait is only due to environmental variation and not at all due to genetic variation. Heritability is also applies only to at the population level. It doesn't apply at the individual level. So you can't estimate heritability from me. You can estimate heritability in the human population, or so because it is a measure of proportion of variance by definition, you require a population to be able to quantify variance. Okay, so it is a characteristic of a population. Another thing that is that heritability changes, and it depends on the environment in which it's measured. It depends on the genetic composition, the population genetic composition of the population. It depends on allele frequencies. It depends on what generation you're in. So heritability can change. You know, at certain generation you can measure heritability, it will be one thing. 10 generations later it may change. And so heritability is not constant at all. It can differ, heritability of a certain trait can be one value in the Dutch population, and it can be a different value in the American population. So it's definitely not constant. it is also important to realize that even if heritability is super high, environment can still influence phenotype in general. And finally, heritability tells you nothing about the difference between populations. So for example, a trait might be significantly heritable, but that's not to say that differences, population level differences in that trait are due to genetics, are due to only to genetic variation. Okay, that's the bit about heritability. How do we identify, or how do we even quantify which loci, how do we identify loci that are quantitative loci? So what we call these loci that influence a quantitative trait, we call them QTL, or quantitative trait loci. And the way we do this is through quantitative trait locus mapping, QTL mapping. And in general, the way this works is that, recall back to the linkage mapping exercises that you did towards the beginning of the semester. This is very similar, where you're going to have what is essentially a linkage map, or you're going to have markers spread throughout a population, or sorry, markers spread throughout the genome. And what you're going to have is you're going to have, say, the trait that you're interested in this case. I think we like to talk about things in terms of height for quantitative genetics because it's a trait that's quantitative, but it's also familiar and easy to visualize. And so if we have tall plants that are, you know, fixed for tallness, and then we have short plants that are fixed for shortness, and we breed them, create an intermediate individual, and then breed that to create an F2, and in the F2 generation we're going to get all possible phenotypes in theory. And the F2 generation is going to reflect all possible combinations of those possible phenotypes, all possible genotypic combinations, and therefore all possible phenotypes. So we're going to use association with different genetic markers. So what we have here, what you're looking at, is two homologous chromosomes. Sorry, a pair of homologous chromosomes. So we have the long chromosome that has A and B on it, and we have the short chromosome that has C and T on it. These are just genetic markers. And, well, A, B, and C are genetic markers. So here we have some hypothetical QTL that's T. And we're trying to identify where T is, and as a way of saying, okay, what are the regions that are associated with plant height in the genome? So we're going to use what's essentially a linkage mapping study, which we call QTL mapping. And we're going to say, okay, we have a number of different, due to recombination, we have a number of different combinations of the A and the B locus alleles together. We have 1 and 1 on the same chromosome. We have 1 and 1, and then 2 and then 1, etc. So we have all possible combinations of these two alleles on the A and B, at the A and B locus, and we have all possible combinations for how they're arranged relative to each other. What we're going to do then is look at, for each locus, and expand this out in your mind to like thousands of loci. For each of thousands of loci, you're going to look at the association between the genotype at that locus and the phenotype, which in this case is plant height. And so for the A locus, we're going to look for an association between the plant genotype at that locus and plant height. And we're not going to see any relationship. There's just going to be a flat line. So essentially, height is the same at all genotypes, and it doesn't really, genotype is not influencing plant height at all at the A locus. Same is true of the B locus. However, if we look at the C locus, we do see a relationship, which is a significant slope between genotype at the C locus and plant height, such that plants with the C2 genotype are taller than plants with a C1 genotype. And so by this pattern, we can conclude that the locus, the QTL, QTL that controls plant height, is located near the C, or at least linked somehow to the C locus. That is the conceptual basis of QTL mapping. Okay, so that's generally, and if we did this across, thousands of loci across the genome, say there's, 10 chromosomes that you can look at, you might get a significant relationship on chromosome 2 and on 4 and on 5, for example. And you may be able to even have enough density of markers on that chromosome to be able to say, not only is it on chromosome 2 or 4 or 5, but it is in this particular region. And so what QTL mapping does is it gets you close to your gene of interest, but it doesn't actually give you the gene because it's based on recombination frequencies, as we talked about when we were talking about linkage. It's not necessarily like super high resolution. All right, so there's another type of, another approach to getting to finding QTL, and that's a genome-wide association study. And the way this works is you do the same thing with this F2 situation, but you basically fully sequence the genome for all of these individuals. And you need a huge populations. You need a huge sample size in order to be able to do this, because statistically, you just because you're doing so many comparisons, you need a much larger sample size. And so you're looking at the association of phenotype and genotype, but you're doing it across the entire genome. What this does is also lets you, because you're doing it at such high resolution, you can get away from using the F2 cross, but you can actually do it on a population level. And so people are using genome-wide association studies to do all, like map genes associated with phenotype for a number of diseases, and using human populations and patient samples. And so it allows you to do these types of studies in organisms and in systems where you can't necessarily breed them very well. So if you have enough data and if you have a large enough sample size, you can do a genome-wide association study just fine. All right, so let's talk about response to selection. So I had mentioned when I gave the breeder's equation, which is h squared equals, oh wait, nope, that's not right. Hold on a second. Sorry, is the response to selection is equal to h squared times s. And so what this means is that response to selection is the difference in mean from the parental generation to the offspring generation under selection. So if you have, so let's say this is a population of plants and you're selecting for plant height and you want to grow really tall plants. So this represents the distribution of the parental generation. You're going to choose a subset of parents to contribute to the next generation. So you're going to not allow anybody else to breed over here. And so you're going to essentially induce artificial selection, and you're going to choose, you're going to choose a population that is towards the higher end to breed. And the difference between the mean of the selected parents to breed and the mean of the generation as a whole, that's your selection coefficient. That's S. Now, if the response to selection is the difference in mean between the offspring of this population, then what happens is that the degree to which breeding this population lets you move your population mean from one generation to the next, that's your response to selection.